Mitochondrial analysis. Mitochondrial DNA is a 1659 base pair circular strand of DNA located in the mitochondria cell and features. It is inherited solely from the mother. Mitochondria DNA in a single cell, on an average, there are 10 raised to 3 mitochondria in a cell. Each mitochondria contains 10 raised to 2 mitochondrial strands copies. A single cell thus contain 10 raised to 5 1 lakh of mitochondria in mammal. It can up to make a less than 1% of total cellular DNA. The basic structures of the mitochondrial DNA consist of a coding region, the entire region other than the blood, okay, which contains sequence of which contains a sequence of 2 ribosomal RNA, 22 transfer RNA, 2 ribosomal RNA, 22 transfer RNA, and 13 proteins, and a non-coding region called the displacement loop or D loop or control region, which is a 969 base pair in length. The control region shows up control region shows variability among the individuals. It is divided into hypervariable region 1, that is base pair 3, and a hypervariable region 2 or HARV2. The variability of the control region between unrelated individual is about 1 to 3%. And into 3 nucleotide out of every. This is responsible for identification. Okay, this is responsible for identification. Numbering of the base pair starts from center and goes clockwise. The position one folds in the middle of the non-coding D loop between individuals. It is efficient in PCR amplification with the intermittent biological material. In addition to the skin, blood, semen, and saliva from which nuclear DNA can be extracted. Mitochondrial DNA can also be extracted from materials which have failed to yield nuclear DNA. Example, teeth. Nuclear DNA, teeth, hair, shaft, bone fragment, even human feces. Okay, it is more robust than nuclear DNA. More robust than nuclear DNA for typing materials due to its higher copy number per cell, and is resistant to extreme environmental conditions. It is more resistant to putrefaction than nuclear DNA. Thus useful in old materials, also useful when material is derived from a dead cellular material such as hair, nail. Its circular nature confirms a protection from action effects on nucleus. Initially, historical material can also be typed. Can be performed on fewer cells due to higher copy number. Mitochondrial DNA can be done from just sperm cells. Contrast about 67 sperm cells are required for STR analysis using conventional nuclear DNA. Haploid nature of mitochondrial DNA makes easier to sequence than diploid nuclear DNA. Case study: mitochondrial DNA analysis. Boston Strangler. That is in 1962 1964, mitochondrial analysis of his supposed victim who had been buried 36 years ago proved that he did not kill her. And next is WTC. Terror attacks at mitochondrial DNA was used to identify dead bodies and body parts. Mitochondrial DNA of its robustness work even the samples are highly degraded as they were in this cases specimens which could not be identified by mitochondrial DNA analysis have been preserved in the hope that a future improvement in DNA technology will provide absolute identification. And also, maritime freezing, because it is carried in mother only, and historical and archaeological investigations, disputed maternity cases also, and human copy number also, forensic value is there. Thank you.